Now, before we start to talk about the gram stain procedure, it's important to have a recap about bacterial classification. Now, we know bacteria are prokaryotic cells, and that means that they have no membrane-bound organelles. Their DNA, their main chromosome, is circular DNA, free in the cytoplasm. It is not enclosed by a nuclear envelope. And we also know that the ribosomes are 70S in the cytoplasm, and they're the sites for protein synthesis. We also know bacteria have a cell wall made of peptidoglycan, and that's in the previous video, part 1. Now, we can classify bacteria based on their shape. And if we see bacteria under the microscope and they appear spherical, like the picture on the top left, so these unicellular bacteria here are spherical or rounded in appearance, we call them cocci. Now, if these bacteria are clumped together, we call them staphylococcus. And one good example of a species of staphylococcus bacteria is Staphylococcus aureus, and we can find this on the skin. Okay, it's naturally found on the skin. Now, if we look down the microscope and we see a slightly different species of bacteria, whereby they are still cocci bacteria, so they're spherical, but this time they're not in clusters, they're actually in chains of cells all attached to each other, as we can see here in the second picture. They're called Streptococcus. So this is a nice example of a Streptococcus species of bacteria. These spherical bacteria are attached together but in a chain formation. Now, sometimes we get bacteria that are Streptococcus, so we know they're going to be spherical, but this time we can see two bacteria are attached together, and we call this actually Diplococcus, this arrangement here. Now, these diplococci, so di means two, so Diplococcus, if they actually attach in a chain like this, we're going to call it Streptococcus, and this is a nice example of Streptococcus pneumoniae, one of the bacterial species that can cause pneumonia in humans. Now, the shape of the bacteria on the right hand side, the Listeria bacteria, we can actually see here they are elongated bacteria, and we call these bacillus. So rod-shaped bacteria are part of the bacillus group of bacteria, and obviously these cells are slightly different shape to the rounded spherical cocci bacteria. And we can see another example of this on the bottom right here. So E. coli or Escherichia coli. These are very elongated cells. They're quite long, and these also form part of the bacillus group of bacteria. Now, if we look at the two sets of pictures, so we can see on the top we've got dark blue cells, and on the bottom we've got pink cells or reddish pink cells. Now, we know bacteria do not appear blue or pink or purple or pink, but this is actually after the gram stain. So the gram stain is a way of differentiating between what we call gram-positive bacteria, which are these purple cells on the top, and gram-negative bacteria, which are these cells on the bottom. And the main difference between gram-positive that stain a purple colour and gram-negative bacteria that stain a pink colour is the makeup of the peptidoglycan cell wall. So all these bacteria on the top row of pictures that are purple in colour will have a thick peptidoglycan cell wall, and all these gram-negative bacteria on the bottom row of pictures will have a thin layer of peptidoglycan plus an outer lipopolysaccharide membrane that surrounds the thin peptidoglycan layer, and we'll see this on the next slide. So here we can see the difference in the cell wall structure of gram-positive bacteria on the left and gram-negative bacteria on the right hand side. So we know the gram-positive bacteria are going to stain purple once we've actually completed the gram stain procedure, and we know that the gram-negative bacteria on the right hand side will stain a reddish pink colour after the gram stain procedure has been finished. Now, it doesn't actually matter what type of bacteria we have. The actual gram stain procedure remains exactly the same. If we do have gram-positive bacteria, obviously they're going to be a purple colour, and if we have gram-negative bacteria, they're going to look different and they're going to be pink. And the difference is because of the nature or the structure of the cell walls. Now we know bacteria are prokaryotic cells. We know they have a peptidoglycan cell wall that sits on the outside of the cell membrane or the plasma membrane. Now in gram-positive bacteria on the left-hand side, if we look at the structure in a bit more detail, we can see here is the cell or the plasma membrane, and this is made of 2 layers of phospholipids. So we call this a phospholipid bilayer. Now we know actually all membranes, whether they be cell membranes, such as this one here, or if it's a eukaryotic cell, organelle membranes inside the cell, they're made of 2 layers of phospholipids called a bilayer. Now this phospholipid bilayer, this plasma membrane of the bacterial cell, would have proteins within it, and they might be intrinsic proteins that span the 2 phospholipid layers, or they might be extrinsic proteins that sit on the outer layer of phospholipids, or even sit within the inner layer of phospholipids. But sitting on the outside of the phospholipid bilayer, the plasma membrane, is a thick layer of peptidoglycan. Now we know peptidoglycan is made from many straight polysaccharide chains, and each polysaccharide chain is made from modified beta-glucose units. And there's actually 2 different types of beta-glucose modified unit. One is called NAG and one is called NAM. So what we actually get is straight polysaccharide chains of slightly modified beta-glucose units called NAG and NAM. The straight polysaccharide chains are actually cross-linked, not by hydrogen bonds in this case, as we'd find in the cellulose cell wall, but actually by short polypeptides. So we get short polypeptides of about 3 to 5 amino acids, and these are the cross links between the straight polysaccharide chains. Now we can see here there's a very thick layer of peptidoglycan on the outside of the cell membrane. Now if we contrast that to the gram-negative cell wall structure, again we can see the plasma membrane or the cell membrane of the bacterial cell, and again this would have intrinsic and extrinsic proteins within it. Now this time there's a very thin layer of peptidoglycan that sits on the outside of the plasma membrane. So it's not this big thick layer on the left, it's actually a very thin layer that you can see here, made of straight polysaccharide chains cross-linked by short peptides. Then there's a little bit of a space, and on the outside of the thin layer of peptidoglycan is an outer lipopolysaccharide layer. Now this outer phospholipid bilayer is rich in these molecules called lipopolysaccharides, and you can actually see the polysaccharide attached to the lipid extends outwards from this membrane. So it's a lipopolysaccharide rich membrane on the outside of the thin layer of peptidoglycan. And it's this difference in cell wall structure that gives rise to the differences in appearance after the gram stain. So this is quite a nice slide to demonstrate 2 different species of bacteria on the same glass slide that have been stained with exactly the same gram stain procedure. Now because we've got these cocci bacteria, actually I'll do that in blue, there you go, here, which are spherical, so we can say cocci bacteria. These are actually gram-positive bacteria because they stain purple. And on the same slide we've got the different species here, and we can see in pink, a reddish pink, we've actually got bacillus this time, and they stain the reddish pink colour. So I'll put the method on the slides in a moment, but we can see here there's a differential stain where some bacteria are purple, they're gram-positive, and some bacteria are pink. Now this slide is a quick overview of the gram stain method, but there's going to be a more comprehensive method on the later slides that we can have a look at in a moment. So this is an overview of the gram stain procedure. Now, the first part of the method is to be able to actually fix your bacteria, whether it be one species of bacteria or two different species of bacteria, to a glass slide. Now, once they've actually been heat fixed to the glass slide, what we can do is add the first dye, and that dye is called crystal violet. And we can see here from the picture, it's actually a purple dye. So we add a couple of drops of crystal violet to our species of bacteria fixed to the glass slide. And this dye actually does bind to the outside of both gram-positive and gram-negative bacteria. So at this point, the crystal violet is actually going to be loosely bound to both species of bacteria, whether they be gram-positive or gram-negative. Now, the second step, once we've actually washed off any kind of loosely bound crystal violet with distilled water, is to add the second solution, which is called Gram's iodine. And what this Gram's iodine does is actually it sticks the crystal violet to the thick peptidoglycan cell wall of gram-positive bacteria. So it acts somewhat like a glue, and we actually call it a mordant. So the loosely bound crystal violet that sits within the thick layer of peptidoglycan of the gram-positive bacteria will actually get chemically linked or bonded to the polysaccharide chains via the addition of Gram's iodine. Now we know because in gram-negative bacteria they've got the outer lipopolysaccharide membrane, the Gram's iodine will not stick the crystal violet to the thin layer of peptidoglycan, because actually the loosely bound crystal violet on the lipopolysaccharide membrane is actually going to get washed off. Now the next step is called the differentiation step, and this is where we actually add alcohol to the glass slide and it will wash the bacteria. Now we know alcohol, like ethanol, is an organic solvent, and it has a couple of jobs. It washes off any loosely bound or non-covalently bound crystal violet. It also dissolves the lipopolysaccharide membrane that sits on the outside of gram-negative bacteria. So by the end of this step, any gram-positive bacteria, like the cocci bacteria we've got in the picture here, they will appear purple after the alcohol wash or the differentiation step. And that's because the crystal violet is covalently bonded to the thick layer of peptidoglycan on the outside of these gram-positive bacteria, and that was as a consequence of the Gram's iodine being added. But any gram-negative bacteria, like these bacillus here, okay, so at this point they're clear, they're not stained with any colour, they're transparent. These are the gram-negative bacteria, and they appear transparent or clear because obviously the crystal violet has not bonded to the peptidoglycan layer. It was actually on the outside of the lipopolysaccharide membrane. That membrane has now been dissolved due to the alcohol wash, and that will expose now a very thin layer of peptidoglycan that was once underneath the lipopolysaccharide membrane. Now we can't just leave the Gram stain at this point here, because if we looked at the bacteria under the 100 times objective lens, we actually wouldn't see the gram-negative bacteria because they're transparent or clear. So what we now need to do is add another dye called a counter stain, and this is a pink dye called safranin. Now safranin also binds to the peptidoglycan layer. So because the thin layer of peptidoglycan has now been exposed on the gram-negative bacteria, the red counterstain safranin will bind to the thin layer of peptidoglycan on gram-negative bacteria, and it will cause them to stain the pink colour. It also does bind to the thick layer of peptidoglycan in the gram-positive bacteria, but because they're already stained a dark purple colour because of the crystal violet that was covalently bonded to them via the application of the Gram's iodine, they're not actually going to change colour. So adding a lighter pink dye to an already purple bacteria, it keeps them a purple colour. So by the end of the Gram stain, we can see any gram-positive bacteria will be stained purple, and any gram-negative bacteria will be stained a pink colour due to the counter stain called safranin. Now we can start to fill in this little activity at the end, and we can just pop in the colours because we know this is going to come up in the exam. So after the addition of crystal violets, any gram-positive bacteria would be a purple colour, and any gram-negative bacteria would also be a purple colour because the crystal violet is loosely bound to both types of bacteria. We then add grams iodine, and we know that covalently bonds the crystal violet to the thick layer of peptidoglycan in gram-positive bacteria, so they will also remain purple. We know gram-negative bacteria are also purple at this point. However, the loosely bound crystal violet on the outside of the lipopolysaccharide membrane is not covalently bonded. So we still see them as purple at this stage, but the next step, that's going to be washed away because it's not covalently bonded to any peptidoglycan. So the third step, the alcohol wash, this is the differentiation step, and this is where we see the first difference. Now, when we add the alcohol, because the crystal violet is covalently bonded to the peptidoglycan layer in gram-positive bacteria, they remain purple after the alcohol wash. Gram-negative bacteria, this time, the crystal violet is washed off with the alcohol, and it's also the outer lipopolysaccharide membrane is dissolved, so that's also removed by the alcohol. So these bacteria now appear colourless. Then we add the counterstain called safranin. We know that's a lighter pink colour, but because the gram-positive bacteria are already purple, that's not going to make much difference. However, this time the safranin binds to the thin layer of peptidoglycan that's now been exposed on the outside of the gram-negative bacteria. So these gram-negative bacteria are going to be red or pink, or reddy pink, at the end of the procedure. So we can follow this procedure through from the start for both gram-positive and gram-negative bacteria. Now we know the first step is the fixation of the bacteria that we'll do in a second, and we call that heat fixation onto the glass slide. We then add the first dye called crystal violet, and we know that's a violet or purple colour. We then add, so I'm going to put these numbers in here, grams iodine. This is a mordant, so it helps to covalently bond the crystal violet to the thick layer of peptidoglycan in gram-positive bacteria. So we're going to see these a purple colour here on the picture. Now, obviously the crystal violet is not covalently bonded to the thin layer of peptidoglycan in the gram-negative bacteria, and that's because the crystal violet at this point is loosely bound to the outside of the lipopolysaccharide membrane. We then decolorize or differentiate with alcohol, such as ethanol, and that is going to wash off any loosely bound crystal violet, but we know gram-positive remain purple because the crystal violet is covalently bonded to the thick layer of peptidoglycan. However, on the right hand side, the gram-negative bacteria now appear clear, and that is because the loosely bound crystal violet has now been washed away, and also the lipopolysaccharide membrane has been dissolved, and that will expose the thin layer of peptidoglycan. The fourth step is the counter stain called safranin. Now we know the pink stain is not going to do much for the gram-positive bacteria because they're already stained quite a dark purple colour, so again they remain purple. But the counter stain safranin now binds to the thin layer of peptidoglycan that's been exposed in the gram-negative bacteria. So we know at the end of this procedure, these are going to be a reddish pink colour. So there we can differentiate between gram-positive and gram-negative bacteria. So this is a more detailed method of carrying out the gram stain. Now it doesn't actually matter what type of bacteria we have, whether they're gram-positive or gram-negative, because the actual method is exactly the same, and we must follow the procedures from step number 1 all the way through to step number 12. So step number 1 at the top then, the first thing we do is to place a drop of distilled water onto a microscope slide. Now we know a microscope slide is a glass slide that we end up putting under the objective lens to look at the bacteria. We then flame sterilize an inoculating loop, so this is a metal loop, and we're going to use that to transfer the bacteria from an agar plate into the drop of distilled water on the microscope slide. Now it's important when you put the metal inoculating loop into a blue Bunsen burner flame, it turns a yellow-orange colour, and that will sterilize the metal loop, but we then must allow that to cool. The third step is then to remove the lid from the agar culture plate, and on the surface of the solid agar, the jelly agar, we'll have our bacteria. So we would have already cultured our bacteria to be able to then transfer some of those bacteria onto the drop of distilled water or into the drop of distilled water. So we take our sterile inoculating loop, we gently touch a colony of bacteria from top of the agar. We then lift the loop away and replace the lid on the culture plate, and we might put that back into the fridge to keep it for next time. We then very carefully rotate the loop with the bacterial sample in the drop of distilled water. So we've now transferred the bacteria from the agar plate into the drop of water. We then re-sterilize the metal inoculating loop, so we can use that later on. We then allow the droplet of water to dry in air, step number 5, and then we very gently pass the glass slide through a blue Bunsen burner flame about 3 times. Now it's important to keep the droplet of water, what was the droplet of water before it dried out, on the upper surface of the glass slide. And we don't want to keep the glass slide in the blue flame because that will crack the glass slide. So we gently pass it through the blue flame 3 to 4 times. When the glass slide is then cooled, we add a few drops of crystal violet, so this is the first purple dye, onto the bacteria, and we let that stand for 1 minute. We then very gently rinse the glass slide with distilled water using a squeezy pipette, and we allow any non-bound crystal violet to wash off and run down the sink. We then cover the slide with grams iodine. So we're going to put 2 drops of grams iodine onto the bacteria, and we'll let that stand for a further 1 minute. It's then important to wash off any crystal violet and grams iodine, but this time using alcohol. So we're going to drip alcohol such as ethanol over the surface of the bacteria until no more blue colour runs out. So the blue colour is really the purple colour of the crystal violet stain. We don't want to add too much alcohol, so maybe a few drops is sufficient for this differentiation step. Now we actually know step number 8 here is really important because this is where the the crystal violet that's stuck to the thick peptidoglycan layer of gram-positive bacteria, because the addition of grams iodine, that will remain. And we know the non-bound crystal violet or the loosely bound crystal violet will be washed off from gram-negative bacteria, and it will also dissolve the lipopolysaccharide membrane on the outside of the gram-negative bacteria, and therefore it will expose the thin layer of peptidoglycan underneath. Step number 9 is to rinse the slide with distilled water again and drain off any excess. We then add the counter stain. Now here it's called phenol fuchsin stain, but this is exactly the same really as saying saffronin. So we're going to put in it saffronin. And this is the ready pink stain called the counter stain. Now we know if we leave that for 1 minute, that will bind to the thin layer of peptidoglycan on the outside of the gram-negative bacteria, and that will cause them to start to stain pink. We then blot the surface of the glass microscope slide very gently using tissue paper. So this is going to take off any excess solution. We then put the microscope slide onto the stage of a light microscope. So this is number 11. But because bacteria are so small, now we know bacteria can range in size from maybe 1 micrometer to 5 micrometer. Sometimes they're a bit bigger. We have to use the 100 times objective lens to do this. If we use the 40 times objective lens, we would not actually be able to see the bacteria because they are so small. Now, what you do is raise the stage and the microscope slide very, very gently. So the glass slide is just underneath the 100 times objective lens. We don't want to touch the objective lens with a microscope slide, so you have to look very carefully when you're doing this. We know we need to put a droplet of oil onto the top of a cover slip that is now on top of the bacteria, and the oil will make a connection between the glass cover slip and the glass objective lens. So this is called oil immersion, and we only do that when we use 100 times objective lens. Now, if you use the fine focus, you'll be able to see hopefully the bacteria on the microscope slide, and we'll be able to differentiate this time between the gram-positive bacteria that are blue, purple, or violet, and the gram-negative bacteria that are reddy pink colour. Now, we just keep repeating this procedure for any different cultures that we might have and we wish to identify in terms of are they gram-positive or gram-negative. So this is quite a nice comparison table to look at the different steps and the different cell wall structures for gram-positive and gram-negative bacteria. So you could possibly pause the video and just have a quick go yourself, just to remind yourself of the key steps, and then we'll look at the answers. So the gram-positive bacteria then, after the gram stain has been added, we know they're going to be a dark purple or violet colour. We try not to use the word blue here just because it is a purpley colour, a slightly darker colour. And we know obviously the gram-negative bacteria are going to be a reddish pink colour. Now we know this is due to the difference in the cell wall structure. So gram-positive bacteria do not have a lipopolysaccharide rich membrane on the outside. They just have a very simple, thick peptidoglycan cell wall. And we know obviously gram-negative bacteria do possess the lipopolysaccharide rich membrane. So we can actually really think of the gram-negative bacteria as having a slightly more complex cell wall structure. Now we know gram-positive bacteria will retain the crystal violet stain, especially after we've added the grams iodine, which is the mordant, and it chemically or covalently bonds the crystal violet to the thick layer of peptidoglycan. But we know gram-negative bacteria do not retain the crystal violet stain. That gets washed off during the differentiation alcohol step. Now we've also covered antibiotic resistance and antibiotic susceptibility before in previous videos. So we know certain antibiotics like penicillin actually only affect gram-positive bacteria. And this is because penicillin actually is an antibiotic that disrupts formation of the crosslinks between, or the short polypeptide crosslinks between the straight polysaccharide chains of peptidoglycan. Now there's an enzyme that's made by the bacterial cell called transpeptidase, and this enzyme actually catalyzes the cross-linking of the straight polysaccharide chains. Now penicillin inhibits this cross-linking enzyme, bacterial enzyme, and that results actually in a very thin, brittle peptidoglycan cell wall in gram-positive bacteria that have been subjected to penicillin treatment. Now penicillin doesn't kill these gram-positive bacteria directly, but what it does, it makes them more susceptible to changes in osmotic pressures. So if these bacteria are placed in a solution, a hypotonic solution that has predominantly water and very few solutes, we know water is going to move by osmosis into the bacterial cell, and that's going to cause the cytoplasm to swell and expand, and that's eventually going to cause the bacterial cell to burst or lyse open because there's no thick peptidoglycan cell wall anymore, just a thin brittle peptidoglycan layer. Now penicillin, because of this effect on the thick peptidoglycan layer, doesn't actually have any effect on gram-negative bacteria, because actually the lipopolysaccharide rich membrane prevents really the entry of the penicillin to a very thin layer of peptidoglycan beneath the outer lipopolysaccharide rich membrane. So examples of bacteria in two categories then. Well, we know Staphylococcus and Streptococcus. We know these are spherical bacteria. We know they've got a thick layer of peptidoglycan, therefore they're going to stain purple. And we know a nice example of gram-negative bacteria is actually this Bacillus rod-shaped Salmonella bacteria. We also know E. coli is gram-negative as well. So some common exam style questions then. We can just have a quick look at these. So Staphylococcus aureus is a gram-positive bacteria. So we can take a highlighter pen and highlight this part of the question, these key terms. And then it says Salmonella enterica are gram-negative bacteria. And then it says, what color would you expect these bacteria to appear after gram staining under the 100 times objective lens? So this is fairly straightforward. Now we know Staphylococcus aureus would appear purple or violet, and we know the Salmonella enterica bacteria would appear a reddish pink color. The second question asks us to suggest what this would tell us about the structure of the cell walls for these two species of bacteria. Well, here we can just put down the information we now know, and we know the gram-positive bacteria will have this thick layer of peptidoglycan on the outside of the plasma membrane. We know peptidoglycan is made from straight polysaccharide chains with NAG and NAM as the subunits that alternate one after the next along the straight polysaccharide chain. These are modified versions of beta-glucose. We know these straight chains are cross-linked by very short peptides, maybe 3 to 5 amino acids long. And then that binds, obviously, the crystal violet stain, and that's added actually covalently by grams iodine. The Salmonella enterica bacteria would have a thin layer of peptidoglycan with an additional outer lipopolysaccharide-rich membrane that actually inhibits the crystal violet from getting to this thin peptidoglycan layer. So this lipopolysaccharide surrounding limits the ability of crystal violet to get to and bind to the thin peptidoglycan layer. Another common question is, what's the differential stage of the gram stain procedure? Well, we now know that this is actually the alcohol wash. Now we can call this a decolorizing step, and we know the purpose is to remove crystal violet, loosely bound crystal violet that has been non-covalently bonded, especially in gram-negative bacteria. Another question is to explain why the two bacteria then look different. So we can have a look at the mark scheme. So we know gram-positive bacterial cells have the thick layer of peptidoglycan, and it's important to say they take up and retain, that really important word, retain the crystal violet. Okay, and this is after the alcohol wash. But gram-negative bacterial cell walls do not take up and retain the crystal violet stain. Now we know gram-negative bacterial cell walls are treated with alcohol, and that then removes the outer lipopolysaccharide-rich membrane that allows the red safranin stain, the counter stain, to then bind to the peptidoglycan layer, the thin layer of peptidoglycan on the outside of the cell membrane of the gram-negative bacteria. So this is quite a nice example of an exam question that actually provided us with a diagram and some additional information that we're going to use to formulate some answers. So it gives us a table, and in the table it does tell us that there's a species of gram-positive bacteria called Clostridium tetani that actually causes tetanus in humans, and a species of gram-negative bacteria called E. coli or Escherichia coli. Now the first question is to complete the table by stating the colour of the different bacterial species. Now the first row tells us that crystal violet has been added as part of the gram stain, and then it says we've completed the wash with grams iodine, which is the mordant that helps to covalently bond the crystal violet dye to the peptidoglycan layer of gram-positive bacteria. And then it tells us that the alcohol wash has been completed, and we know the alcohol wash is the differentiation step. So we can work out actually what the bacteria would look like for gram-positive bacteria. Now we know they're going to be a purple-violet colour, and that's because the crystal violet dye is bonded covalently to the thick peptidoglycan layer, and the alcohol is not going to disturb that crystal violet, so it doesn't wash it off from the gram-positive bacteria. However, we do know that any loosely bound crystal violet will be washed off by the alcohol, because the grams iodine has no effect on gram-negative bacteria, because the crystal violet cannot be covalently bonded to the peptidoglycan layer, because it can't actually get through the lipopolysaccharide-rich membrane. Now we also know the alcohol wash, alcohol is the organic solvent, and it does dissolve phospholipids and lipopolysaccharide membranes. So it removes the outer lipopolysaccharide membrane to expose the thin layer of peptidoglycan. So E. coli, the gram-negative bacteria, will actually be colourless at the end of the differentiation alcohol wash. So here we're going to say it remains colourless. Now the next row at the bottom says what would happen when safranin has been added as the last part of the gram stain. Now we know gram-positive bacteria are a dark purple colour, so adding a lighter reddish pink counter stain actually doesn't have much of an effect, so they remain purple. So it must be the same colour as the answer that we put up here. Now we don't actually say crystal violet's a blue colour, it's a darker purple or violet colour. Now we know E. coli is gram-negative bacteria when we add safranin. Well, this safranin now combines to the very thin layer of peptidoglycan, and that's going to make them appear a reddish pink colour. Now the diagram on the right hand side in figure 1 is the structure of a gram-negative bacterium like E. coli in the table. And question B says name structures A and B in figure 1 and then state the function of both. Now we know, if we start with B actually at the bottom here, we know this is the phospholipid bilayer because we can see the polar head and 2 fatty acid tails, and we know that there's 2 layers of phospholipids, so we know this must be the cell membrane. So we can also call that the plasma membrane of the bacterium. And you can actually see here the intrinsic proteins that span the width of the phospholipid bilayer, and we can see some extrinsic proteins here in the outside layer of phospholipids, and further down we might have some here, for example, in the inner layer of phospholipids, there'll be extrinsic proteins. Therefore, we now know that sitting on the outside of the cell membrane or the plasma membrane is a very thin layer of peptidoglycan, represented by this picture here. And outside that, because it's gram-negative, this double membrane here would be the lipopolysaccharide rich outer membrane. So this here is the lipopolysaccharide membrane. Now actually the lipopolysaccharides, just to complete the diagram, are these modified lipids on the outside, and the inner layer of this double membrane, or this bilayer, these are phospholipids here, the regular phospholipids that we see in a phospholipid bilayer. So these molecules on the outside are actually the lipopolysaccharides. So A is the thin layer of peptidoglycan. Sometimes you might see this written as murin, but we prefer the term peptidoglycan. And what's the function? Well, it helps give the bacterial cell structural support, and it prevents osmotic pressure from a hypotonic external solution causing damage or bursting of the bacterial cell. So even though it is a thin layer of peptidoglycan, it does give some structural integrity to the bacterial cell and prevents them bursting open if they do find themselves in a hypotonic external solution. And we know hypotonic means that the water potential of the external solution is less negative. So it's much nearer to water. So it's higher or much less negative compared to inside the bacterial cell in the cytoplasm, which would be more negative. So we have a more negative water potential value inside the cell. Now we know B is the plasma membrane, otherwise known as the cell membrane, or you could say phospholipid bilayer. And we know the function of a phospholipid bilayer, or a cell membrane here, is to be selectively permeable. So we know certain molecules can actually get into the bacterial cell by simple diffusion if they are non-polar or not charged. So non-polar or not charged, they can simply diffuse into the cell across the phospholipid bilayer. Molecules like oxygen can diffuse in, or CO2 can diffuse out by simple diffusion. We know also that if they are charged or polar molecules, like glucose for example, they will need to go through a specific carrier protein, such as this intrinsic protein that spans the width of the phospholipid bilayer. So that's why we can say that the cell membrane is selectively permeable to certain molecules. It will only allow certain molecules into the bacterial cell as long as there are specific carrier or channel proteins for those molecules or ions.